Today, we will be examining Oswald, Avery, Colin, MacLeod, and Maclyn McCarty's experiment of enzyme digest of Griffith's transformation factor. As you're probably guessing, this is, in fact, a further study on Griffith's study conducted back in 1928. So we can understand the context of our experiment, let's go visit our dead friend, Frederick Griffith. Hello, Mr. Griffith. Nice to meet you.
Hello. Here's my card, strange person from the future. I'd like to start off by saying I am a super smart scientist, and I did some pretty cool stuff back in my day. Would you like to hear about it?
Actually, not really.
Okay, so back in 1928, while attempting to create a vaccine for pneumonia, I was studying Streptococcus something or other, I don't really remember, it's not that important. Anyways, a bacterium that causes pneumonia in mammals. As I was saying, that's terrible tea, I was using two strains, one pathogenic and one non-pathogenic. And whenever I heated the pathogenic bacteria to the point of death and mixed the cell remains with the living bacteria of the non-pathogenic strain, some of the living cells became pathogenic. Yes, indeed.
Oh.
I found this newly acquired trait of pathogenicity was inherited by all the descendants of the transformed bacteria.
Drinking the bad tea.
Keeps the pneumonia away. And this meant that some of the chemical component of the dead pathogenic cells caused this heritable change. Yet the identity of the substance was unknown. I dubbed this phenomenon, I dubbed it transformation, a new word for the 1900s. Now defined as a change in genotype and phenotype due to the assimilation of external DNA by a cell. Cool, right? I know. Yeah. Would you like to hear about my other experiment? You see, I was.
Yeah, all right. Okay, thank you so much for your time. We really have to get moving. Bye-bye. So now we've learned about Griffith's experiment. Wasn't that fun? Now let's get to what we're really here for today. Next on our interviewing list is the dynamic trio themselves. However, only Oswald Avery was able to make it today. Oswald, you there?
Hi there. Nice to meet you.
So, what famous stuff did you do, Mr. Celebrity?
Well, aside from my amazing social life I had with all my friends, no, I'm not talking about my textbooks.
Oh.
My two friends and I basically redefined how we look at information today. But it's no biggie.
Wow, that sounds so interesting. Can you tell me a little more?
Yeah, sure thing. So, back in 1944, a lot of scientists were under the assumption that the unknown substance found in Griffith's experiment, have you heard of him?
Yeah, sure.
Was a protein. However, I really wanted to prove or disprove the assumptions made. To do so, I had to choose a bacteria to examine. Specifically, we chose to examine Pneumococcus. However, we did not want to just examine the entire, but only the active portion of the bacterium.
Well, how do you only look at that part?
It's not really that interesting. You should really ask me about my...
Seriously, dude?
Okay. So, basically, we isolated the active protein and then extracted it to examine it. To do this, we first killed the bacteria with heat and extracted the saline-soluble components. From there, the protein was precipitated out using chloroform and the polysaccharide capsules were hydrolyzed by an enzyme. Finally, the active portion remained, and that was able to be precipitated out with alcohol fractionation, and that could easily be removed using a stirring rod.
Wow, that's a mouthful. So what did you do from there, once you had the active portion?
Well, we proceed to examine it by looking at the chemical composition of the active portion. Carbon, nitrogen, hydrogen, and phosphorus were all found. Moreover, these findings aligned with the composition of DNA instead of RNA, protein, or anything other cell component responsible for the transformation.
Wow, so you're like a super genius.
Well, in addition to this, we tested trypsin, chromatrypsin, and ribonuclease, enzymes that basically break apart proteins or RNA, and found that none of these affected transformation. However, whenever we tested deoxyribonucleic depolymerase, an enzyme that breaks down DNA, the transformation power was destroyed.
Dang, so it basically further verified the conclusions you came to?
Yeah, you can put it like that.
Great, thanks so much for your time. This was super interesting.
Thank you very much. I'm surprised you really didn't ask for my number.
Never.
Well, maybe my book?
Goodbye. These conclusions that the three scientists discovered were not widely accepted until 1952, where Hershey and Chase used radioactive isotopes to prove that it was primarily DNA rather than protein that entered bacteria upon an infection. Today, I will leave you with rare video footage from the Scientific Archives of America of Avery, McCarty, and MacLeod performing their experiment. Thanks for watching, and remember, it's a great day to be a knight.
Avery, thank goodness you're here. McCarty was just about to start the experiment without you.
Yep, I'm here. Begin the experiment. Alright, let me write down the data on this one. Alright, extract the liquid. Who knows, maybe we'll change the face of science one day.
Sure, definitely. This is weird. It doesn't actually look like protein. These four elements are...
I recognize them. I see traces of nitrogen, phosphorus, carbon, and oxygen.
Oh my god, it's DNA!
Wait a minute. This isn't protein.
Oh my god,
I've seen this before.
Not protein.
it's DNA.
That looks really familiar. This might...